Chapter 21, congenital and genetic disorders. And this is just an introduction that we have 23 pairs of chromosomes in the human cells. Each nucleus will have that number, 23 pairs. 22 of them are called the autosomes, and these are just the regular chromosomes that are controlling the whole entire body. The last one, which is number 23, this can be XX in females and XY in males. Do you remember this? So we have 23 what? 23 pairs. Okay? So it's 46. And one pair of sex chromosomes, males XY, females it's XX. Now there is something called karyotype and genotype. What is that? Karyotype is the visual representations of the chromosomes. They are arranged in order of size and shape. And I will show you a picture of that karyotype. So karyotype, you're looking at how the chromosomes looks like. And they will be arranged in that manner, size and shape. How does it look like? And this will be used to diagnose chromosomal disorders. Give me that karyotype, I'll look at it, and I will see if there is one of the chromosomes that doesn't look okay. Does it look normal? Okay? This is for the karyotype. Phenotype, we will see it later on. This is how these chromosomes are presenting. We will see that later on. Meiosis and mitosis, I think I talked about this. Mitosis is the way all cells in the body reproduce. Meiosis, on the other hand, is only in the sex cells. The only type of cells that you have meiosis. And by the way, meios is Greek for reduction. Meios, translation, reduction. So I have this 46 chromosomes. I'm going to split them into 23. So you start with one cell that have 46. You have two cells. Each one have 23. Isn't this is half of the number? Are you reducing the number? This is where meiosis came from, just to understand it. Mitosis is coming from mitos, and mitos is threads. Do you remember the threads attached to the chromosomes, split them apart? So this is, if you wanted to understand where meiosis and mitosis came from. So meiosis, what are the only cells that have meiosis? 6 cells. Ova and sperms. This is the karyotype. Look at it. So this is the typical, how it looks like. So I will look at it, it should look like this, and see how it's arranged in size. Size is bigger here, smaller. So size and shape is how I arrange it. And I always take the chromosomes from you and compare it to this. I compare all the 23. Does it look okay or not? If it does not look okay, I can diagnose any genetic abnormalities. So genotype and phenotype. Genotype is, we're talking about the actual genes. Okay? All 22 pair of chromosome, they have the same characteristics. If I have chromosome number 3 and chromosome number 8 and chromosome number 21, all of them will carry all sets of genes, the same thing. So each one, it's identical. All of them will have the same set of genes. Not number 23. 23 is different from person to person, males or females. Okay? So you have the same set of cells in your cells? Yes. But not all of them will be expressed. Okay? Some chromosomes have genes that will be expressed and some others will not be expressed. Phenotype, on the other hand, this is the expression of the genes. So like the gene that's determining that your hair will be brown, I call that genotype. Is that clear? The gene that's determining this. But your hair is brown. This is the phenotype. Did you get the idea? Genotype, phenotype. For the same characteristic, yes. Genotype, genes having criteria. Phenotype, how is it expressed? It's expressed that your hair is actually brown. This is the phenotype. How you look like, the appearance. They have a project that's called the Human Genome Project, and this is mapping the nucleotide sequence to identify the genes of each human chromosome. So they have this genome project, and they use it to compare different chromosomes and know what's happening. Congenital disorders, we have different types of disorders in genetics, but generally speaking, disorders present at birth. These are the congenital disorders that show up right at birth. And this will have something inherited or something developmental. So it's something that you are born with it, or something that will take some time to develop. And usually it manifests in the neonatal period. Neonate is a neonate, just born, right? Disorder that are inherited can be caused by one of the three. It can be a single gene expression. You have a problem in the gene number 18, for example. Gene number 21. You have an issue in this one specific chromosome. Sometimes you have chromosomal defects. You're losing one, you're having additional one, and so on. Sometimes it's polygenic, but our focus on the single gene one, okay, which is the Mendelian one. Are you familiar with the Mendelian inheritance and genes? Okay, so you do have different things, but the main focus on the single gene. So the single gene, which is called the Mendelian disorders, these are traits that are controlled by one set of alleles. What's the alleles? Alleles are the genes that are located in the same site on a pair of homologous chromosomes. Match the function. So I have this chromosome. Okay? I have this allele here, and the other allele is on the other side of the chromosome. Okay? These are called alleles. It's pair. One here and one in the other strand. Okay? So each trait is controlled by one set of alleles. Those two together, this allele and this allele, those two together are determining what's your hair color, for example. Are you going to be short? Are you going to be long? And so on. Your color of the skin, whatever. Everything is determined by one set of alleles. Chromosomal anomalies, usually the problem is happening in meiosis. Why not mitosis? Because mitosis is happening in the normal cells. You're not going to inherit it. If something happened in the normal cells, you as a person will get a problem. But if this issue happened during meiosis, meaning sperm or ova, this is what's going to go to a next generation, right? To the offsprings. Something like non-disjunction or translocation. What does that mean? Non-disjunction is, let's say if I have this chromosome, this one, right? So now you are trying to do meiosis, and you're going to reduce these cells. So you separate them, it become like this, okay? And here's another one, and then you're going to put them back together, like this, okay? So if you, during doing that, this part, leave this chromosome and go to this one. This is called translocation. A piece from this chromosome, translocate it, go to another chromosome. So you're supposed to do it perfectly. I have this pair, separate them, make a complementary from this one, make a complementary from this one, everything is going fine. Sometimes, while you're doing this, a piece cut and go put itself on another one. This is called translocation. Non-disjunction is, while you're doing this, you're separating them. So this chromosome is okay, okay? This one is separating, and it's in the process of reproduction. This one did not separate. So the next step is, you will have this and a third one, and this will be only one. Did you get the idea? It didn't do disjunction. It's supposed to do like this. Disjunction, double this, everything is okay. If you're trying to do this with one chromosome, it did not detach, and the other one did. So now let's exchange the chromosomes. I give you one, you give me one. You didn't do the disjunction in the first place. So one of them will come to this group, the one that didn't do disjunction, and the other one will stay by itself, just like this. It will become 1 and 3. Translocation versus...
Wait, so no disjunction is it when a piece breaks off, but it's when like a whole side goes to another chromosome?
Translocation, part, leave, and go to another spot. Okay. Translocated itself. The non-disjunction is they did not separate from each other. and then one from you're supposed to do like this. Here's one chromosome, okay? And here's the other chromosome. They're supposed to do like this, and this one is supposed to do like this, and then they exchange, okay? If this does not separate, it's not going to exchange. This one separated. So I give you one chromosome, give me yours. No, I did not do the disjunction. Okay, non-disjunction. So this is one, and this will end up having the 3. Teratogenic agent. Teratogenic. Genic means to generate. Terato means congenital. So these are agents that generate congenital anomalies. Okay? And this is something that happened during embryo or fetal development. Okay? Early in pregnancy, it's called embryo. And then the embryo ultimately become a fetus. There is a difference here. Okay? Is it the first 2 months or is it after that? So at the beginning you call it an embryo. It's not the same. In the beginning you call it an embryo. Later on you call it a fetus. It's not the same. So this is one teratogenic. So it's not like the chromosomal abnormalities that we talked about. It's actually something happened during embryo or during the fetus. Like, example, this mom got a German measles, for example, and she's pregnant. Does she have an embryo or fetus? It depends on the age, if it is less than 2 months or after. Okay? So German measles, what's going to happen? The cells are growing and reproducing, and the virus is going to invade and cause teratogenic effect. You know, of course, that the pregnant ladies should restrict taking certain things, a lot of things, right? You don't take this. If you have to, then take Advil, for example, right? Can I give this treatment for hormonal therapy? Can I get that? No. You're not supposed to take any one of those. So if you start to take a medication that you're not supposed to take, it can affect the embryo or the fetus. The next part is the multifactorial. So what's a multifactorial? Multifactorial is another issue that have an environmental part of it. It's not just the chromosomes. It's a combination of something wrong happening in the chromosomes plus environmental factors. I call that multifactorial. And these are the ones that will have familial inheritance. Other types of congenital development, something like premature, like he's born at 5 months, just happened. Difficult labor and delivery. The size of the head is too big and it's going through, take more time. It can cause cerebral palsy, for example. Did you hear about this before? This baby is taking more time to get out of the cervix, stayed for some time, compression, and the baby was born with paralysis, cerebral palsy, right? So these are other congenital disorders. So if we talk about the single gene disorder, this is the Mendelian one, and this is the main focus, okay? So these genetic disorders, which is the inheritance ones, it can be recessive, dominant, or X-linked, and we will discuss this. So we're talking about single gene, one gene, whatever it is, number 21, for example, whatever it is, not number 23, okay, any other gene. You're having an issue with this one, can be recessive dominant, or if it's in number 23, that will be the X-linked one. So you're talking about single one that's controlling like the gene that's supposed to give you the ability to see colors. If this gene is affected, you get color blindness, for example. Some of them will give you systemic effects like cystic fibrosis, Tay-Sachs, phenylketonuria, PKU. These are other things that will give you systemic effects. Cystic fibrosis, for example, if you inherited that gene, defective gene, it's a defective gene, one gene that have a defect. This one gene is supposed to give you the ability to produce the surfactant. Are we familiar with this? So there is one gene, whatever the number is, it's supposed to give you the ability to make enough surfactant. Okay? You're having a defect on that single gene, single chromosome. So you do not produce that surfactant. you end up having something like cystic fibrosis, for example. You have inability to secrete fluids, and this will cause problems all over the body, including the pancreas and other things. Phenylketonuria. What's a phenylketonuria? Phenylketonuria simply, we have different amino acids, right? One of them is called phenylalanine. You're supposed to, this is coded in the genes, you're supposed to have the ability to break down or to work on these amino acids and change it to something else. Use it. If you have this issue, you cannot use phenylalanine. It's going to accumulate, you eat it. You're supposed to get rid of it. Use it. Use it in proteins, break it down, whatever, right? You're eating something to use it. You can't use it because the gene that will enabling you to use it is defective. So phenylalanine accumulates and start to appear in the blood, start to appear in the urine, and this is called phenylketonuria. So we have autosomal dominant disorders, and we will discuss what it means. Something like adult polycystic kidney, Huntington, familiar hypercholesterolemia, and Marfan syndrome. By the way, you have to memorize these examples. You have to know it. So if I tell you Huntington chorea, what is that? Is that a single gene or multi-gene? You have to tell me this is a single gene. Is that dominant or recessive or X-linked? It's dominant. If you don't know this, you're losing points. You have to memorize all these examples. So number one is autosomal recessive. When we say autosomal, whether it's recessive or dominant, we're talking about a chromosome in the first 22. Is that clear? You're having an issue with the first 22 chromosomes, not number 23. If it's a number 23, this is the 6th chromosome, which is X-linked. Are we following or not? I have an issue with the chromosomes. Which chromosomes? Is it from 1 to 22? Or is it number 23? 1 to 22, I call it autosomal. If it's 23, I call it the X-linked. So for autosomal recessive disorders, both parents, they will pass an allele, and you have to have a combination to be affected. Male, if that's the case, males and females are affected equally, and this will not be the case when we talk about X-linked, right? If you're female, don't you have XX, right? So this will be different than a male who's having XY, right? We will see the details. If this is a heterozygote child, there will be no clinical signs, and he will just be a carrier. And look at this. This is extremely important. You will see, you don't have to see this exactly, but you will see description of this. Is that clear? You will see this Punnett square, not necessarily the whole square. I mean, it's not easy to put this in the form of question, but I can describe it to you. Like I tell you, you have two parents, okay? The father and the mother. One of them is carrier, and the other one is carrier. Can you tell me the chances? How many will be normal? How many will be carrier? How many will be affected? Are we following or not? Okay. So if here is a scenario. I don't have to show you this. I can describe it to you. I tell you this is an autosomal recessive, or I can tell you this is Tay-Sachs. Tay-Sachs, is that autosomal recessive? And that's why I told you to memorize these examples. You have to. I don't have to ask you, this is an autosomal recessive. Can you tell me how many normal and how many? I don't have to say that. I can tell you Tay-Sachs, cystic fibrosis. So the first thing to come to your mind, Tay-Sachs, cystic fibrosis, this is autosomal recessive, and you remember this in your mind. You don't have to see it. Okay? So can you give me the chances? Yes. Here is C, uppercase C, lowercase c, uppercase C, lowercase c. So this is autosomal recessive. Okay? This is the one that's defective. One of the alleles. And you get the alleles, remember that you get from the male and from the female. You get one from here and one from there. We know that, right? You have 23, it will become 46. How did you get it 46? I get 23 from the male and I get 23 from the female, right? So each one of those will give me one allele. What's the allele? I'll send each one of those. This and this. This is coming from male and this is coming from female. You have both of them. One of them is affected. Is that the case? So if I have the good one, this is one chance. The good one from the male and the good one from the female. You're completely normal, right? This is an example of getting the bad one from the male or getting the bad one from the female. It's a chance, right? You never know the child which one it's going to be. Or it can be this with this. So one affected and one not normal, you have a mix. If you have a mix, do you show the disease? No. The disease will, what autosomal recessive means? You have to have the two alleles in order for the disorder to show up. Is that clear?
Yeah.
You have to have both of them. What if I have one and one? One good and one bad, like this. This is bad and this is good. You're not normal. You're not normal. You're a carrier, just like this. So are you getting an issue? No, I'm not getting an issue, but I can pass it to the next generation. Males and females would be equally, yes.
So if you have parents and both your parents were carriers, then the child will be affected?
No, it's 50% will be carrier, 25% will be normal, 25 there's chances. You don't know. Like this is a carrier male and a carrier female. Do you know exactly which allele are you getting? You never know. It's random. Okay? Am I getting from my dad the good one and the good one from my mom? Maybe. So you'll be normal. Am I getting one and one? You'll be carrier. Am I getting the two bads? I don't know. Yes.
What is PKU?
Phenylketonuria. Phenylalanine. Phenylketonuria. I think it's right here.
Yeah. Wait, what is it actually?
Phenylalanine, it's one of the amino acids, accumulate in the blood, show up in the urine.
Oh, okay.
Because you cannot use it. Tay-Sachs, we don't have to know the details. I'm just giving you an idea. Tay-Sachs is a problem with storing the metabolites. So you're not supposed to, metabolites are not supposed to be stored within the cells. This is an abnormality, like the neurons. In the neurons, you're not supposed to have like a lot of fat inside the neuron. It's an glycogen. I'm not supposed to have that much. So it's a metabolite that's abnormal storage. You don't have to know the details. If you know the names, I'm just giving you an idea if you're wondering what it is. But you have to know. You have to know, is it recessive or dominant, and what are the chances? So we get one normal, one affected, and two carriers. So the chances are, so you're telling me, okay doctor, here there is a couple, male and female, both of them are carriers. Can you tell me the child that we will be born, what are the chances? I will tell you, I cannot tell for sure, but I can give you this. 25% he will be normal. 25% he will be affected. 50% he's carrier. Did you get this? And you will see description. I don't have to give you that exact thing. Here is another one example. One is affected and the other one is normal. All of them will be carriers. Look at it. So in your brain you should say, okay, if one is totally normal and the other one is totally affected, none will be affected. All 100% carriers. So if you're asking me what are the chances, I will tell you all your kids will be affected anyway. Because you have to take one from here and one from here, right? Can you take two from the female, from the mom? No. There is an ovum, this is a sperm. Did you get the idea? These are the examples that has to be memorized. Autosomal dominant. In autosomal dominant, there is nothing called carrier. Even if you have one, you get the disease. That's why it's called dominant. Okay? So one allele is enough. There is nothing called carrier. You have the disease, you don't have the disease. The carrier is only in the recessive one. Usually these conditions appear early in life, but in autosomal dominant, some of these will be evident later on. So this is born. Can you tell me if he's having this issue or not? Huntington chorea. Can you tell me if my baby is having Huntington chorea or not? I look at the baby and he looked normal to me. But maybe in 5 years, 10 years, he will develop, show up later. And this is something for the autosomal dominant. Okay? So it's kind of delayed. This is the Punnet's square. You have to memorize these examples. You forget it, you lose it. I don't have to tell you autosomal dominant. I can tell you adult polycystic kidney, for example. And if that's the case, look at this. And you have to make the scenarios in your brain. Having one affected and one normal. What's going to happen? 50% affected, 50% normal. How about if I have one affected and the other one is also affected? What's going to happen here? 3 will be affected. But look at this. This is affected with both of them. And this is, can you call this as a carrier?
No.
Even though it's mixed, right? There is no carrier in autosomal dominant. And these are the examples. I changed these slides a lot, by the way. So this is different than the original slides. I put some stuff on it, like I put these here, just to make it easier for you. So you see examples and you see the square, so you can tell. And I added and removed some stuff just to make it a good source of studying. Okay. Next is X length. Yes.
Is that in the list or is it in like the reading? This PowerPoint that you changed?
This is the one that I uploaded in the Canvas.
Okay. Under like
on the modules.
On the list. Okay.
The original wasn't there, but I advise you to do this.
Okay.
I changed it to give you like all that you need to know, or most of it at least, 90-something%. Okay, so X length. X length mean in one of the 6 chromosomes, right? X. So X length can be recessive or dominant as well. If it is recessive, it's in X chromosomes anyway, not in the Y. It's called X length, so it's in X chromosomes, not the Y. So if this is a heterogeneous male, and he's having a Y, it will show up. If it is a female, it will not show up. It will be a carrier. Here is why. Here is a female who's having an issue in the X chromosome. Okay? If this X chromosome go to a male and Y, it will show up. Why? Because you don't have one that's normal to match it and to correct it, right? If you have one normal and one abnormal, you will be carrier. But you cannot be carrier as a male because you have only one X chromosome. But if you're taking one, if you're female, both of them are X. One of them is affected and the other one is not affected. The one that's not affected is going to balance. Did you get the idea? So you don't get it, but you're a carrier. And only if you have both of them, you will be affected as female. Did you get the idea here? Is that clear? If you're female, you have a chance. Even if you get one. Did I get one? Did I get two? If you get one, you'll be a carrier. If I get two, you'll be affected. In males, it's different. In males, you'll be affected if you get one, because you don't have another X chromosome that will be affected. That will, balance it for you. It can skip generations, which was not the case in the autosomal. It's not skip. And here are the examples. Duchenne and hemophilia. Color blindness is number 3. And I put the examples here to make it easy for you. So here the situation is different. Here is the female, the mother, and this is the father. Okay? And here you have female that got both of them okay. Male have X and Y, right? I'm a female. I get one X from here and one X from here. Both of them are okay. I'm fine. I'm normal. Okay? Here is another female. I got one from the mom. That's bad. It's recessive. And one from the father. I'll be a carrier. Okay? If you got one normal X and you have the Y, you will be normal. If you are male and you got one, you will be abnormal. So if this is a mother that's carrier and a father that's okay, do you have a chance of getting an affected female? You can see a question like this. A carrier mother and a normal father, does the females have the chance to be affected? Of course not. He's normal. So one of the X's would be fine anyway, right? He's normal. One of the X's will be fine. So I get one fine. You get me another one fine, I'll be totally normal. One fine and one affected, I'll be carrier. There is no chance to get an affected female. Did you get the idea? If the mother is a carrier and the father is affected, yes, this is where you can see females that are actually affected. On the other hand, if this is dominant, both of them will be affected. I don't have to have, because if it's recessive, I have to have both of them to show up, right? Or one if you are a male. If this is dominant, one is enough. So males and females will be equal. It doesn't change. It's not different. And the example of that is Fragile X syndrome. One more time, you have to remember all these examples. I don't have to tell you this is X-linked dominant. I can ask you this question, but I can say Fragile X chromosome, for example. And this is the most common genetic cause of cognitive defects. The most common, for your information, what's cognitive means? Intellectual, right? Intellectual, like memory and all that stuff. So what's the most common in all over the world? What's the most common? It's Alzheimer's. Okay? But I'm asking you here, not what's the most common. What's the most common? Genetic one. That's coming from genes. Alzheimer's is not a genetic one. Right? So what's the most common one? It's Fragile X. This is the most common genetic type. So these are all the examples. They just combined it. And there is one thing here that you should know. Sickle cell anemia is one of the recessive. Okay? But there is something called sickle cell trait. Did you hear about this? Sickle cell trait is you not have the sickle cell anemia. You're just a carrier. So and this is recessive. So I don't have it, right? No, you're carrier. So this is heterozygous. We call it incomplete dominance. What do you mean by that? Here is what I mean. If this is something recessive, okay? I have one bad and one good. I call that carrier, right? What will happen to you? Absolutely nothing. I don't have the disease. I'm carrier. I will pass it to the offsprings. I have absolutely no issues. But is that the case here? No. Here is a carrier. This is a unique thing. Okay? This is a carrier. He's having one good and one bad. He's a carrier. So, of course, you mean that he's a carrier. He's totally fine. No, he's not totally fine. The people with sickle cell trait, if you expose them to severe hypoxia, very low, they will get the sickling. Do you have a disease or not? Do I have it or I don't have it? In the middle. I can't say you're totally free and you don't have the disease, you're a carrier. So carrier, and because this is incomplete dominance, it's not exactly recessive, it's incomplete dominance. So this is an example that's important to know. And for your information, those people with so what's sickle cell trait? It's a carrier. Do you have sickle cell anemia? No, you're a carrier. Is that clear? If you have sickle cell anemia, you get sickling all the time. But this is not the case. I have sickle cell trait, I'm totally fine. Unless there is severe hypoxia, I see the sickling. Is that clear? And there is something that's weird, something unusual. This type of blood cannot be invaded by malaria. Like they can go to any country that everybody's having malaria, they don't get malaria. Because their red blood cells is bad for malaria. Malaria cannot survive in these cases.
Is that anyone with sickle cell or just a sickle cell trait?
Sickle cell trait. Okay, so this is something unique that I wanted to mention about sickle cell trait. And chromosomal disorders that we should discuss, Down syndrome, which is trisomy 21, and trisomy, what's tri means? 3. So this is an disjunction that I talked about, right? Number 21 doesn't have 2. It's supposed to be 2. You have 3. So you end up having Down syndrome, non-disjunction, sometimes dislocation. Turner syndrome is XO. What's O means? 0. You don't have. So you just have one X. So you got one X from the mom, for example, nothing from the dad. It's an abnormality. And this is called Turner syndrome. These are the females that will be very short and infertile. Turner. Klinefelter, on the other hand, XXY. They have, this is a male that's having an additional X. He will be a huge person and infertile. So Turner, Klinefelter, and then the Down syndrome. And Down syndrome is one of the mental, people who have mental issues. So which chromosome is affected? 21. And what's happening to that chromosomes? It's 3. Instead of 2, you have 3. Did you understand this? This. So this is happening in number 21. Okay? It's a common one that's affecting both physical and mental development. Both of them will be affected in Down syndrome. Down syndrome increase with the maternal age, and that's why they always say, if you want to get pregnant, for females, if you want to get pregnant early. Don't wait after 40s. You have a very good chance. 50, after 50, very big chance of having, you don't have to get it. You have a lot more chances. Okay? So it goes with the age. And there is a screening that's called the triple or the quad screening that's available besides the first trimester test. And this is something that should be done automatically. Any female who is pregnant is supposed to do that. We're supposed to give you the triple or quad, and maybe also the first trimester screening test to know if you have an issue or not. You can diagnose this early. So what's a triple? 3 things together. I'm going to measure the AFP, the HCG, and estriol. These 3. If you add inhibin A, it's the 4th one. You call it the quad. So is it a triple or is it a quad? You need to remember these things. Okay. What should I do for screening in the first trimester? You're supposed to do ultrasound, also known as ultrasonography, right? Ultrasound and maternal blood screening. If you do that screening, you can tell if she's having an issue or not. The ultrasound is specifically for structural abnormalities. It does not tell me anything about the genes. Structural means, yes?
Where it says maternal blood screening, does it mean take the mother's blood, or do you have to have like amniocentesis for it?
No, from the blood of the mom.
Oh.
It's just a screening. The amniocentesis is something else. I'm talking about the blood of the mom. I can take it and I measure these 3. If it is high, I can tell you that you have a good chance of having trisomy 21, which is Down syndrome. Okay? You need to remember that ultrasound or ultrasonography, which is the same thing, is for structural abnormalities. So I can see that the baby is very short. His hand is small. His face is small. Structures. Something wrong with the structure, with the physical characteristics of the fetus. On the other hand, you can use amniocentesis or chorionic villi sampling to confirm the diagnosis. So if I take the blood and I see this high, what should I do now? I can, if it is high, I go to the next step, which is not good because it's very aggressive. I go to the baby itself. Okay, I take, the amniotic fluid surrounding the fetus or the embryo. I can take a sample from that, and I call that the amniocentesis, or a sample of the amniotic fluid, which is surrounding the fetus, to act as a cushion and for nutrients as well. Or I can take sample from the chorionic villi, which is the beginning of the placenta. I can take it's not going to much affect the baby, but this is called an aggressive type of screening, an aggressive test. Okay? Invasive test. Blood sample from the mom is not invasive. Are we okay so far? Yes.
So the only time you would do an amniocentesis is if the maternal blood screening came back
Yes.
normal?
Yes, because this is the easy one. Or if you have very good chance, like she's 50, just do it. You have to do it anyway. Characteristics are a lot, but the ones that I put underline are the most important ones. Okay, so this is what you're not supposed to forget. The head will be small, the face will be round, the facial profile is kind of flat, like not everything that's fully grown. The tongue is large and protruding outside the mouth. I will show you a picture. Heart will be affected, short stature, cognitive and intellectual function will be impaired, sexual development will be uncompleted. If this is a male, it will be infertile. If this is a female, it will have low rate of conception. There is a difference here. If this is a male with Down syndrome, he's infertile. There is no way he will make conception. If this is a female, conception will be lower. Look at this picture. Round face, flat facial expressions, small eye. If the tongue is outside, if you open his mouth, you see the tongue is big and protruding out. Small eye. The next is the multifactorial, and this is of course have to do with, beside the genetic part, you have an environmental aspect, and you have to know these examples. Okay, cleft palate, congenital hip dislocation, congenital heart disease, type 2 diabetes mellitus. So type 2 diabetes mellitus is that genetic? There is a genetic factor, but you have to have an environmental factor as well. Developmental disorders. During development, during the fetal time or embryo, you're exposed to something that you're not supposed to be exposed to. Like this is a pregnant mom who's taking certain drugs, exposed to chemicals. So if you're pregnant and you're working in a chemical factory, stop working. You cannot be exposed to these chemicals. Radiation, don't go through the airport a couple of times if you're pregnant. Infection, specifically torch. And this is something that's automatically screened. These are infections, virus, that if the mom, pregnant mom, got one of those, she might develop congenital anomalies in the baby. It's not like common colds or something. Like common cold, the mom is sick, that's it, right? But this is different. So this is toxoplasmosis, others, which is IV, hepatitis, varicella, parvovirus. The R is for rubella. C is for cytomegalovirus, H for herpes simplex. Sometimes they call it the torch, sometimes we call the torches, S. If that's the case, you're adding the S, which is the syphilis. And yes, you need to remember these. I can ask you what torch means. Okay? During the first two months of life, which is during embryo, you can be exposed to something of the teratogens, and we talked about this. Teratogens. If you're exposed to one of the teratogens during the first 2 months, you will have impaired organogenesis. What's organogenesis means? Yes, exactly. The organs are developing, and you have an issue with that. So the liver is developing, and then stop. If you're exposed to any of those, they will not have metabolic abnormalities. If that's the case, you don't have metabolic abnormalities, but the organogenesis will be affected. Organogenesis. Cocaine, for example, if this is a pregnant mom that is exposed to cocaine during pregnancy, she might end up having a premature birth, respiratory problem, or SIDS, which is sudden infant death syndrome. Of course, any pregnant lady should not use cocaine, or even if she's not pregnant, right? If you're exposed to any one of those chemicals, torch, one of the torch, anything, if that happened during this period of time, the zygote and embryo will die, like in the first 2 weeks. If it happened after that, up to 8 weeks, you have major abnormalities. If you're exposed after that, you will have less issues. So is it worse to be exposed early or later in life? Early is the worst. Early is the worst. If it is early, the zygote will die completely, death. After that, you will have abnormalities, not death, but you'll have abnormalities. After that, like after 3 months, 4th, 5th, 6th month. So anything after, you will have minor abnormalities. So the early exposure is the worst of all.
So like if a girl's pregnant and she does drugs, and let's say you don't find out until, a few weeks, until you've been carrying for a couple weeks. So before you actually find out that the baby's growing in your stomach or it's forming, that is that time frame? Or after you find out? Because before you find out, the baby's there.
Finding out is not going to change the fact.
Right.
If it happened within the first 2 weeks, it will lead to death. Okay? If it happened in after that, but before 3 months, there will be major abnormalities. Whether you find out or not, this is something else. When are you exposed? During the first 2 weeks, after the 2 weeks, but before 3 months, or after that?
Like just thinking about women in different, whatever their lifestyles are, and they do drugs, let's say. I mean, you don't know for 3 weeks
that she is pregnant.
You're even pregnant. So like
least.
all those babies,
Yeah, it will.
some of them have lived.
It will happen. It will happen. Okay. So there are a lot of tests that are available there, but pregnant ladies above 35 is the most important, and you have to do the screening. Blood test, we talked about this. In utero, we talked about amniocentesis and chorionic villi, and I mentioned that amniocentesis is for prenatal diagnosis, and you get a sample of the amniotic fluid, and this is the most invasive. You can do blood, you can do ultrasound, it's not very invasive. The most invasive is amniocentesis, because you're introducing a needle inside and taking a sample. You might go a little bit more and you injure the baby, and it will lead to issues. Others can be in the neonate. After birth, you can see something like you can do some test for phenylketonuria or blood samples. You can do that as well. Genetic engineering is you are engineering the genes. You take the genes and you play with it. You can isolate, you can make copy, you can transplant, you can take it and put it like in a bacteria or something, multiply. So it's good for examining these genes and changing the natures. Like we hear about orange that's like this, right? How did they do it? They take the genes and they engineered it, which is called the genetically modified. So, and they can also change the DNA sequence. And the goal of that is to make sure that the alleles is good. Okay? This is already used for animals and already used for plants, but they are trying to apply that for human being as well. Gene therapy. Gene therapy is basically to know the genes and the protein that's responsible for the condition. So let me see what's happening. It's a gene 21. Let me find out, and then I can tailor the treatment depending on that. And before it's actually start to express itself. So the gene is going to lead to protein synthesis. Before doing that, I can know the gene and I know how it's going to be expressed. Cancer they are working on developing drugs for cancer by doing the gene therapy. Genetic screening and DNA test. If this person is at risk, you can do screening, but the problem is screening is not always the solution because it's very expensive. They cannot let's do PSA for all males. That will cost like billions of dollars. So it doesn't go like this. I have to be specific with more higher risk people. Designer drugs. I think we're almost done. Designer drugs. Proteomic research. What's the proteomic research? Proteomyces, these are proteomic research is research concerning the proteomics. What are the proteomics? You are examining these with their functions. What are proteomics? These are the proteins resulting from activation of a gene. So here is one gene. I'm activating this gene, and I see the result of that, the protein that's synthesized. When I study this is called the proteomics. And this is regarding the gene expression, the protein that's going to be expressed, what's the metabolic pathway. So this is designer drugs. You're designing the drugs depending on the proteomics that you are studying. So this will reduce the unwanted side effects. So basically, designer depend on the proteomics, which is the protein expression, to tailor a medication specified for this person. Okay, this is the bottom line of this one. So this is for chapter 21.